So what I'm going to just talk about is really the mouse as a model. I think John gave a great, the ethical, and posed a lot of those questions. And this will give you some, hopefully a little bit more of that reality of what the work's like when we're working in an animal facility. This is an early drawing of the work that we had, and the trust invested some 100 million, I think it was around 12, 15 years ago. Having done the genome project in this area, they then moved on to invest in the information that had come out of it. And one of those parts was this section at the back here, which is the RSF, the Research Support Facility. One of the biggest in the country, one of the most productive in the country. And I'll try and explain to you why that's actually a good concept when you're talking about the use of animals, and try and tie into some of the 3 R's and the way we think about things and how we can reduce that burden on a large production. So these are our little friends. They somewhat our heroes, I think, as well, in the way they work. One of the questions is, I suppose, to ask is why the mouse, and why is it so important? And probably the biggest thing in terms of genomics is the fact it's very close to our genome. So they're around about 96, 98% homologous, so the same genetically as we are. So if you go back in time, around about 100 million years ago, they diverged away from us. So there's a lot of conservation of the genomes between the two species. So that gives us something of an opportunity then to look at the mouse to kind of infer the function of a gene to the human state. So in terms of that, I'm going to cover those few areas. Just to give you a little bit of, about our background and the areas, and very, very similar to the Babraham in the types of research that we're doing. Human genetics, looking at development, looking at variants in the diseases that affect us as a population. Obvious ones, cancer, we're all susceptible to that. There's some great work going on at the moment discovering the interactions of genes. So more we understand, the more difficult and the more complex it becomes, because we're having to look at multiple mutations within the animals and then back to the humans. And then we go on things like malaria, neglected diseases. So because of the funding we get from the Wellcome Trust, we're very fortunate, but we can look at these diseases that aren't maybe so popular, and it gives us that opportunity. The Research Support Facility has an output, so we try to make sure that we're, the animals that we use, we talked about the benefit to mankind, but we're always balancing a cost to the animal, and that's the thing that we're trying to ensure, that there's a value to the information that we produce. We term them model organisms. The mouse and the fish are those animals. To us, they're a well-known organism. We understand them, we understand the genomes of them, and we're able to apply our research to that. We supply to the scientific community both live mice and fish, as well as frozen embryos and sperm, and I'll go on to that a little bit later on. Experimental data, training, publications on our own work, and actually welfare initiatives. So the 3 R's, we're looking at ways to reduce the burden on the animal at all times. So when you was having a look around the virtual world, one of the things we saw was, I think Paul would have explained, the embryo superovulation. That allows us to manipulate. This is kind of the classic scene you'll see on the telly when they're talking about IVF and things like that. Here, what they're actually doing is injecting into the pronucleus. You can see that little rounded area there with the nucleus sitting inside. So they're they're injecting DNA into there. There's other forms and techniques. Some of them can produce what we call a chimera. So these are made up of 2 sets of cells that both contribute to the animal, one being from the embryo itself and the other being manipulated stem cells that we apply. That allows us then to breed animals and bring them into being and look at that mutation, and then to pull them together. And even now we're beginning to look at even broader genomes and interactions. So in this instance, this is a germ-free animal, and what we're looking at here is the effect of the microbiota on our own genomes. So the modern animal facility, as we break it up, centred around the mouse being probably the most popular of models, and certainly the heaviest used one within the UK. And there's all these aspects to it. Some of the key ones are the husbandry, the underlying work that we do, genotyping, the phenotyping type work, even movement of animals. But the reality of this is, for us in the facility, is this what it looks like? There's a huge amount going on. So it's not just simply caring for animals and looking after them. We're interacting, we're providing data, working with the sequences of these animals, supplying them out, talking with, looking at histology, working in collaboration with other groups. So there's a massive amount of work going on to unravel those genomes of the mouse. We're quite stuck by one thing though, and it's the animal. It actually controls the work that we do. And one of the things that the mouse lays down to us is it has this very, very controlled, if you like, sort of generations as you produce them. So the gestation period is 3 weeks. The pre-wean up to the point of weaning is 3 weeks, and then they mature, and they'll sexually mature in another 3 weeks. So for us, we can actually begin to look at the way we operate in there and base our decisions on progressing a model around that. And then from there, we can try and tie that into the experimental work that we're trying to do. We look at that and we try to find opportunities to reduce the number of animals that we're working with. And here we can look at all of the different stages of production and make key decisions on them within a time frame. And this actually has an interaction of 6 different groups at any one point in time where we're trying to pull together that. Don't have to know the detail of this, it's just to show you the complexity. So for us, when we're breeding them, that very early stage of that first injection is up here. But to get to the results that we're after, the immune challenges, the recessive lethality as an example, these are the things that we're trying to do. So we're constantly breeding this under demand to try and meet the requirements of the science that's going on around us. This was actually quite an interesting one. We did this with the NC3Rs as a challenge. Mathematically, it's actually very difficult to see if you can find, we talked about reduction, is there a way we could reduce at any of these stages the number of animals we're doing? And it turns out this is probably one of the most complex mathematical questions that have ever been posed to them. So it's not quite as straightforward as we might imagine. So one of the things we do see, lethality, subviability and viable. So this is a line, a mouse model. Now, if we bring together 2 deletions of a particular gene, then they could be lethal. And what we see is actually fairly representative of the human state as well, is that around 25% of those are actually lethal. So will cause development issues that mean you fail to have a pup in our case, or potentially a baby being born. This one is probably our most concerning one in terms of the welfare of the animal, because subviable means that by definition they live to a certain point. So we then have to apply a lot of strong care and welfare to those animals, so we take very close attention to them. So we wrap all that together, and actually one of the things we challenge ourselves is to try and do the work in as few animals as possible. And this is an interesting thing for us. We developed a database, it's used here as well at the Babraham Institute, and part of that was looking at how we could overview all of the colonies we're producing. And you can see here in 2007 there was around 600 colonies in the research support facility, accounting for some 11,000 cages, and on average that meant around about 19. Over the years, going this way, we've monitored that and actually looked at a more effective way, and you can see the cage colonies are coming down, and it sort of brings us to this point about a year ago where we were around about that 12, so we've reduced the number of cages down significantly. That turns out it's all to do with better management, quality management, making sure, and I showed you that effective management, how we could bring that down and reduce that to a level where we only produce the number of animals we need to produce for the experiments we're running. The other big part for us is actually that we distribute to the world, and it literally is the world. We go all over the place with our animals, and we have to sort of balance the fact that you're sending a live animal and the potential stress to that animal versus the production. So in terms of production, by us doing it in the most efficient way, we can offset that. And it actually means if everybody had to go and produce those lines again in the different parts of the world, you'd have had to have used another 450,000 plus mice now to produce them. So it's a way of us, if you like, justifying the work we do and the way we do it at scale. So I'm going to move on very quickly now to talk about the animals themselves and the care and the welfare. So this is one for you two. What I'm going to do, we're going to have a look at the animal itself. Imagine you're now the person, the animal technician, and you've got to try and tell me what you're seeing with that animal. So, would you say that's normal or abnormal? Yeah.
It's hopping more.
Looks like a bunny rabbit, doesn't it, the way it's moving around the arena. What you've actually got here is the spine is fused slightly, so the gait is abnormal. The animal itself is actually very hyperactive, and it develops cataracts. So you can see it moves around, it's happily moving around, but it's something we then have to look at in terms of that animal and is it normal or abnormal. Now, we begin to now apply different conditions to that animal. We start to think, okay, so is that, and this isn't particularly severe, but if it had an issue with writing and coming up, could it actually eat and feed itself? So could it get to the food? Could it water itself? Is there a problem with that animal? So it shouldn't be so difficult. You've seen it there, but of course what you're trying to look at is base this against a normal mouse. So you see one running around out in the field, you see it for a brief fleeting second. What we're actually able to do is see that animal literally from the word go, its birth, all the way through to the end of its life. That means that when we're looking at them, we're applying not only the normal condition of the animal, but then beginning to look at the factors that we apply to it. So things like the genetic manipulation, harmful phenotypes, procedural burden, so the experimental burden that we place upon it, and then very much the cumulative effect of those. And this is a challenge that's been thrown to us under both EU law and our own law. So we have to make that call on the animal and then report that out so that the public can actually understand what we're doing and how we're doing it and what the burden to the animal is. We talked about the cost benefit, and that's what we're trying to show here. So a normal mouse, what is a normal mouse? So for us, we talk about things as being outbred and inbred. So the outbred ones, you don't control the genetics. The inbred you do. They literally are bred over 20 generations until they're almost identical. Some of the normal things that we expect to see are things like the number of litters that are in there, the average litter size, age at sort of sexual maturity. So these are things we can map. But when you start moving into inbred strains, all of this begins to alter. That then has an application on the animal itself in terms of what we're likely to see in terms of a phenotype. And here, just 3 different models. These are all inbred strains, the black 6, C3H there. You can see the types of different phenotypes and traits that we're likely to see in each of these. So you can imagine if you were trying to do age-related hearing loss, the black cyst might actually be a useful model to you. Equally, if you were trying to look at alcohol preference, it looks like this little baby actually quite likes it. So you've got to be quite careful with some of these things. So what we do see is actually there's a really diverse range of phenotypes that we're seeing within the animals, and trying to understand those is hugely important. And you can see here Mism was highly phenotypic. There was a hell of a lot going on in that animal. Altered immune system within there, body weight, it was runted, it was quite a small animal, problems with the spine in there was a lot. Yet this model here, absolutely nothing going on, and that's not so surprising. The model itself is just a genetic component for other models. We don't expect to see anything going on in there. But what we're trying to do is actually sort of get that information in a controlled manner and present that in a way that it's actually helpful and useful. At certain points in the development of the animal, we can look at things like, is there a milk spot? So the actual animal when it's born is completely hairless, and you can actually see whether it's been feeding, and it literally is like a little white milk spot in there. So you can see things like that. You're expecting to see the eyes closed. As the animal ages, things like the eyes begin to open, and by P14 days of age, you'd expect to see them open. So when we see something like this with the SPNS2, you can see the eyes were open at birth. So that's abnormal to us. We actually kind of expected this with this one. There were some indications on it, but what we were doing was looking to observe that, and was there something we could gain in an advantage when we were breeding and managing these animals. This is another example of a model here, SRC. If you look at the normal wild type animal, you can see how the lower jaw and incisors extended out. And this one, they're quite runted and stunted in there. What that actually meant was that as a young animal, we saw this runting in there, the missing teeth, and the technicians observed that. And because of that, we actually said to ourselves, there's an issue with breeding these animals, and it was actually wrong for us to continue breeding, and so we stopped that, because you would have meant that animals of that what we'd call a homozygous then didn't have the ability to survive and thrive. So we altered the way that we actually approached our research. And we do that, don't worry about the contents of this, but this is a lot goes on, so we can make very rapid decisions, and we use this as a way to make that decision. If we see that issue, as we did with the SRC, we would then be able to very quickly get these groups of people, the technicians, the NACWO, so NACWO is a welfare officer looking after the animal, the experimental team, and then each of these within this group can make a decision to do something with that animal and hopefully protect it. And what that means for us is that this early intervention helps us to prevent welfare issues within the animal. So just some examples here that we've managed these animals, brought them down. We have a pipeline that runs for 16 weeks, but in each of these instances, you can see here, we've terminated that early. We've still managed to go on because of the way we've approached it to get valuable information that we can then pass out to the scientific community to inform their science. And by doing that, we've met our obligation to the Wellcome Trust in terms of publication, and we've also met our obligation to the animal in terms of the ethics and the welfare of it. So just a very quick, brief overview of the type of work we're doing, but just a summary, the mouse, as we said, has got a very comparable genome to our own. That's why we use it. There are a diverse range of phenotypes, and that's, if you like, the interesting but the challenging part for us as a community, as a group, trying to look after those and make sure that the welfare issues are managed properly. There are about 20,000 plus coding genes. There's a lot of work to do. When we finish doing all of those, then we'll start looking at them in combination, and we're already beginning to do that. So that in itself becomes even more of a challenge. Complexity of the programs and the need for this harm-benefit justification underpins everything we do. We're constantly challenging ourselves with that, and as John said, you know, if you go to any animal facility, the first thing they'll say to you is the 3 R's. That's the thing we're trying to ensure, and that welfare then underpins everything that we do in the way we work and operate. And that's just some of our beasties. There you go, thank you.
James, thank you very much. It's very interesting, certainly for me. I've not heard James speak before, and it's nice to see how his explanation of the work at the Wellcome Genome Campus matches what I already know about the facilities here at Babraham, which I know more about obviously than the Wellcome. And I hope that connection has come through to you guys as teachers, and then that too, I think, or I hope, also is matched by John's opening talk, which explains how rewarding it can be to discuss these things with your students. I hope that the combination of those three things has given you food for thought and information about how you can bring this subject up with your students. Hopefully you'll have picked up the resources from the back of the classroom. There are, in at least the plastic bags, some of the web links to find those resources, which we've used all of them very successfully in school visits, and hopefully you'll find the same sort of use. Is there anything that you guys have already done in your schools which you could share with us at this stage? Or would you like more information about what you can do in conjunction with either the Babraham Institute, the Wellcome Genome Campus, or indeed understanding animal research in order to bring the subject more to life in your own schools? If nothing at the moment, certainly if you'd like to email me, Fran, Steve, after the event, and we'll, anything we can do to help, we're happy to do so. But it's, as John said, what we really like to be able to do with schools is that we're not trying to convert anyone to our way of thinking. We're not trying to hide anything. We're trying to make sure that students have a greater understanding, so that whether they've got an opinion that matches ours or not, they have an educated opinion. That's all we ask, and we make that very clear with any of the students that we speak to. So any questions you have after the event, we're happy to speak to you or provide you more information.
Thank you very much.
Any questions for James or John for either the other?
It's just that we talk about animals, but we talk a lot about vertebrates, and I'm guessing, I have no idea, like, you talk about fruit fly at some point. Is there any regulation on how to use fruit flies?
There is no regulation for invertebrates. That doesn't mean that there's no welfare concerns, and there's some interesting studies going into what the welfare conditions, what would mean good welfare for certain invertebrate species. But it's unregulated from a legal point of view. It's not included under ASPA, which is the act which controls.
So it's actually just vertebrates?
It's vertebrates, and with one exception of the cephalopods, the octopuses, the squids, the cuttlefish. But otherwise, it's only the law only covers vertebrate species. I often say to classes, there's one invertebrate species included. I guarantee someone will say snake. Snake, which is more or less a backbone with a head on it, which is, but in some way, I mean, you think.
And you also mentioned that we're not allowed to use great ape in UK nor in the EU. Since when?
Well, certainly since ASPA, which is 1986, and I believe that there was you'll know more about this than I do, guys, probably, but that there was there had been a sort of like industry's the wrong word, but they had ceased to use them since some time before then, but I can't remember exactly when that was. But it's certainly since 1986.
Ever since I've been in, there's been no use of great apes. So.
Yeah, I mean, it's legal to use them in other jurisdictions like the States, but they are falling out of use even there.
Something else, there's a lot of sort of interest in like how CRISPR technology is being used. Is it having an impact on your work with mice?
Huge impact. So do you know about CRISPR? Mm-hmm. Yeah. So in terms of it as a technology, it's making a lot of the accessibility, the speed at which we can target the genome much more accessible to us. It actually means from a welfare and from an animal numbers point of view, we're reducing down the numbers that we have to use also. That's a good side to it. But there's some questions over it as a technology. So is it actually as effective and as targeted? Do we see off-target effects? The evidence at the moment is no, we're not. But it's a continual challenge to us as a community. There are some technologies that it can't use. So we didn't go into too much of it, but there's a group that's called conditional models where you can basically switch on and off the gene. They're quite complex constructs that we put into stem cells, but at the moment it's not amenable to CRISPR. Once that comes online, then we'll have a really powerful tool that will allow us to access into the genome of the mouse, and ultimately I suspect that will transfer into human use as well.
And I would imagine that in turn, just as genetic testing has been in the curriculum for some time, the ethical implications of things like CRISPR will start to move into the curriculum, probably from A level, and then it work its way downwards. And it'll be interesting to see at what stage it becomes mainstream enough to be included in the curriculum. I know some teachers are very interested in it at the moment, and one of our events next term should be on CRISPR.
Yeah, the October one will be on CRISPR.
Yes, so watch this space if you want to find more, even more about CRISPR than you may have found out at the last Bioscience LITE. But it will be interesting to see how these ethical considerations come into the curriculum as well as the actual techniques themselves.
I think one of the first questions it's going to throw up when it gets to humans eventually, but it's actually, it now allows you to access other mammalian models as well. So whereas the mouse is probably the workhorse of genetics at the moment, you'll begin to see other models being used as well. So they're already looking into rats, as for example, so as an alternative model. And there's actually a lot of common sense to that, because we do a lot of work in the mouse, but actually, for argument's sake, if you were dealing with asthma, a rabbit is a much better model for looking at that. So it makes it more appropriate to the research question you're asking. So.
How many strains, because is the aim to create a knockout for every single gene in the human genome?
Yeah.
So how many have we produced to date, and
so
how far?
think at the last skills, well, from our point of view, we've done well over 1,000 of the 20,000 coding genes, and I think there's some very large consortia that are bringing that together. We've created stem cells for over 16,000 of them, so we can then start working through that. And I believe, don't quote me on this, but I think the figure's up around about 3,000 to 4,000 models that have been produced. With those, there's a lot more models, but the reason this is so important is we do it in what we call a controlled background. So the little fella in the middle is what we call a C57 Black 6N. So it's a very particular inbred strain that we use, which means that we control and standardise the genetic background. So the only mutation occurring, or the only difference, theoretically, is that one change that we've applied to the animal. So a lot of work to do. Some genes will be inaccessible, so we've already begun to find out there's certain ones that we don't quite understand what the genetics of it, but we just cannot target into those. So there will be a few that are so critical there's just nothing you can do with them.
Well, again, thank you very much, John and James, and thank you very much to our audience for coming along. I hope you've enjoyed it.
Thanks for
There's information in the bags, there's some feedback forms in your bags, which we would like your feedback, please, and look out for information about the next Biosciences.